Welcome back. This is part 3 of chapter 7, the second video. The first part of chapter 7 we did together before spring break. So now we're going to move on to our next mechanism of evolution, which is non-random mating. So think back to the assumptions of Hardy-Weinberg, and we looked at the other mechanisms that were outlined by Hardy-Weinberg before, which was selection, drift, mutation, migration. So one of the other assumptions of Hardy-Weinberg was that there was random mating. So if we relax that assumption and we have non-random mating, that can result in natural selection or evolution. Okay, so in this slide, what we're seeing is essentially a very extreme case of non-random mating. What you have here is a population of 3 different genotypes, and what we're going to do is we're going to make this population become hermaphroditic. So basically everyone self-fertilizes. So A1 mates with itself, A1 mates with itself, A2 mates with itself. So extreme case of non-random mating. And what's going to happen, we can kind of see down here on part B, that what's going to happen is initially it was in Hardy-Weinberg equilibrium. And then every generation we are losing heterozygotes, because remember, when heterozygotes mate with themselves, when an A1 mates with an A1 half their kids are A1 but half their kids are 1/4 of their kids are A1 and half, I'm sorry, 1/4 are A2 So every generation we're going to lose half of our A1 and dump them into the 2 homozygote categories. So every generation we're losing half of those and we're giving them to the other groups. So we're going to lose our heterozygotes and we're going to increase our homozygosity. So selfing is kind of an extreme condition, right? What if instead of mating with oneself, they mated with relatives? Well, the results are going to be very, very similar in that situation. It will lead to a loss of heterozygosity and an increase in homozygotes. Just will not be as drastic or extreme as what we saw here. This table just shows us a general model of what happens to the frequency of the 3 different types of genotypes when you have selfing. And so as we saw before, every generation you are decreasing your heterozygotes by 1/2 each time, and you're adding those each into your 2 homozygote groups. So it's just a general model of what we saw before. These are some data, some empirical data from sea otters. And we're looking at 2 different populations here. We're looking at California and Alaska populations of sea otters. And the California population of sea otters was harvested very heavily. And it went through a bottleneck where there were probably fewer than 50 individuals. And of course, anytime you go through a bottleneck like that, you end up losing a lot of your genetic diversity. Because of just random genetic drift causing some alleles and certain combinations to be lost. So what they did here is they went in and they looked at, as you can see here, 31 different loci for these different otters, and essentially looked at what percentage of them were, of those loci, were heterozygotes or heterozygous versus homozygous, and they compared that number to what would be expected. And so they used the expected under Hardy-Weinberg. So if it was a completely random mating situation, what would we get? And then what do we actually see? And so you can see in the California population where the heterozygotes expected were 7.2, but those that were actually observed were 4.6. So there's a pretty substantial drop in the heterozygosity in the California population. In the Alaska population, it still is a little bit lower than what we would have expected, but not as low as we saw in the California population. So that drop in the population size essentially makes it so the individuals are actually breeding with other individuals that are mated to them, or I'm sorry, related to them, because that's what happens with small populations, and we're seeing that loss in heterozygosity that comes along with that. This figure is trying to show how you would calculate an inbreeding coefficient, which we can also think of as a degree of relatedness. This figure from the book is a bit confusing. So think about your cousin. So let's look at this focal individual. Think about your cousin. You receive half your genes from your parent, and then that parent shares half their genes with their parent, right? So this is kind of going back this way. So 1/2 and 1/2. And then because we're talking about your cousin, this common grandparent here, between this is your mom and your uncle basically, this common parent here shares half of their genes with that individual, and then they give half of their genes to this individual. So if we follow this back, we end up with 1/2 times 1/2 times 1/2 if we follow each of those sections, and we end up with 1/8. So if you were to look at you and your siblings, you share half your genes with mom and half your genes with your dad, and so do they. So on average, you and your siblings are also 50% related on average. So you just trace back basically the ancestry to look at the degree of relatedness. Now, when individuals mate with others who share a lot of the same genes, we call it inbreeding. And it may not necessarily affect allele frequencies, but it does affect genotype frequencies, where we actually get fewer heterozygotes and more homozygotes. So think about many genetic diseases. A lot of them are recessive. So as long as you don't end up with 2 copies, you're okay. You don't have the disease. But if you get 2 copies, you do have the disease. Well, if you think about inbreeding, you're more likely to end up with 2 copies because you're mating with individuals that share similar genetic backgrounds. So you're more likely to get those 2 copies. So since those genes are often associated with disease, inbreeding gives rise to lower fitness, which we call inbreeding depression. Some data from humans that illustrate inbreeding depression. So what we can see here is on our y-axis we have the mortality rate among children of first cousins. So when first cousins had kids together, this was the mortality rate of those children. And then on, that was the y-axis. Then on the x-axis we have the mortality rate of children from unrelated parents. And so data points that are on the line, there would be no difference. So whether your parents were related to one another or not wouldn't matter. The fact that the vast majority of these data points are actually above that line says that the mortality rate among first cousins, this on the y-axis here, is much higher relatively than mortality rate among unrelated individuals. And so again, that mortality is a measure of fitness. We see reduced fitness when we have inbreeding and individuals mating with other individuals that are related to them. This figure shows a similar situation. We have data from flowering plants. What we see up at the top is the coefficient of inbreeding. And so as this number gets higher, there's more and more inbreeding going on. And so what we're seeing is a lot of different ways that that coefficient of inbreeding can change different aspects of the plant's biology. How large it is when it flowers, the emergence time, seed weight, seed set, all of those things are things that are likely to influence the overall fitness of the plant. The data we see here are from birds. And in this particular case, what we're seeing is the coefficient of inbreeding. As that coefficient increases, so we get more and more inbreeding, the number of eggs from a clutch of eggs that fails to hatch increases almost exponentially here. So the more and more inbred the parents are, the more likely those eggs are to not hatch. So that's clearly, by definition, inbreeding depression. So now we can talk a little bit about the Florida panther. Unfortunately, the Florida panther is sort of the poster child for inbreeding depression because they are highly inbred. Essentially, the population became very, very small. There's a very low population size that got down to, I don't know, maybe 20 or so individuals in the wild. It's recovered a little bit now. I think it's a little bit higher than that. But once you go through a bottleneck like that and you lose that diversity, you really don't get it back unless you introduce new genes into the gene pool. So if you just take 20 individuals and now you grow your population from those 20, you're still going to have very high inbreeding. There's no new genes coming into that population. So the population is highly inbred. You have a lot of alleles that are fixed. You have very low heterozygosity, and there's a lot of inbreeding depression. So this explains a little bit of what has happened in the Florida panthers. So again, we see this very small population, very little gene flow coming in because of the isolation of the Florida panther. And so genetic drift definitely results in some of that random fixation that we see and the reduced heterozygosity. So what ends up happening is overall, if we were to take the average fitness of a panther from one of these highly inbred populations, the average fitness is very low. One of the things that we often see as we look at these kinds of situations where we have a lot of inbreeding, and you see this term here that says genetic load. The genetic load is the accumulation of these harmful recessive alleles. So these disease-causing alleles, they accumulate and they build up, and so you get more and more copies of these harmful alleles, and that, of course, makes it so that you exacerbate the inbreeding depression. There's also, it mentions here also, a synergistic interaction between three things: mutation, population size, and genetic drift. And they call this synergistic interaction a mutational meltdown, because basically what happens is you have a really small population size. So in a big population, when you have a mutation, it might be 1 in 100,000 individuals, so it's almost non-existent, right? The frequency is so low. But if you have 20 individuals and now one of them has a mutation, that's a pretty high frequency, right? Now it's maybe 5% carries this mutation. So in a big population, mutation doesn't really have big of an effect, but in a small population it does. Add to that, small populations also have the effect of genetic drift, where you have this random changing of allele frequencies just by accidental sampling, and those things all together make it so that this population is very much in crisis. The bottom here talks a little bit about what are some of the actual phenotypic effects that we see as a result of inbreeding depression in Florida panthers. So they have a lot of heart defects, very low sperm count. They're very susceptible to infection. They have really, really high miscarriage rate. Very few of their cubs go to term. So there's very low birth rate. And then those that are born, there's a very, very high infant mortality. So their success rate, their reproductive success rate, and their overall mortality is very, very low. Well, the mortality rate is high and their success rate is low. These are some data showing inbreeding depression data for different cats, and we see the Florida panthers, and we also see some other cats for comparison. And so one of the things you're seeing is on this, when we compare lions, cheetahs, pumas, some of the other cats, we see that the Florida panther has one of the lowest amount of heterozygosities that we see in these populations. So there's, for a vast number of their loci, they are fixed. So they no longer have any heterozygotes, they're fixed. And of course, if they're fixed for harmful alleles, and those are expressed in the recessive, that's why we're seeing a lot of that inbreeding depression. So we see very, very low heterozygosity. The data that we see here look at some attempts to show what has happened in the Florida population, the Florida panther population. If you notice, from 1986 to around 1996, the population heterozygosity drastically decreased. And then they started recovering the population a little bit, and we see that heterozygosity has come up a little bit. The heterozygosity can only come up so much if the population tanks, because again, you only have so much to work with if you don't bring in genes from the outside. So raising the population up is going to help that heterozygosity a little bit, but for loci that are already fixed, then there's no getting heterozygosity in a fixed locus. So it's only going to help those that have not yet reached fixation. If we look over here on the graph on the right, what it's looking at here is we see these bottom two graphs where they were looking at the average heterozygosity and how it affects the annual survival of the panthers. And so we're seeing pure Florida panthers in the red dots. We're seeing admixed panthers, so where populations kind of were combined with one another, but still, you know, the panther populations essentially in the blue dots. And so we're seeing that if you increase heterozygosity, you clearly do increase the survival a little bit. Clearly really low heterozygosity has a lower survivorship here and a lot more variable survivorship. But look at what happens up here with these green data points. Those are our F1 panthers. So what they did there is they took basically Florida panthers and they brought in panthers from other populations and they had offspring. And so that first generation of those offspring of those hybrids are these F1 panthers. And so in essence, if you were to take these two, the red and the blue data points here, those populations, and rather than look at them individually, you would allow them to cross with one another. You could then see how that actually increases their heterozygosity quite a bit, and it also drastically increased their actual survival. So increasing that heterozygosity by bringing in different genes is definitely the way to go for some of these populations. This last slide just shows us a video link, which is a video that essentially shows you an animation of how two forces of selection and drift work in the same population. So if you have both drift and selection, what is the outcome of that? And so that simulation will show you that. And that is the end of chapter 7.